Hello students. Today we are going to discuss some important questions for ICSE 2020-25 biology. In this video, we will discuss the questions according to their chapters. I have set some questions in for each chapter, and one by one we will discuss each chapter. So our first chapter is structure of chromosomes, cell cycle and cell division. First of all, this chapter holds great marks in our exam. So, what are the important questions for this chapter? First question: in which stage of cell cycle DNA replication takes place? As we know, DNA replication consisting of two phases: interphase and mitotic phase. Interphase, and the second one is mitotic phase. During interphase, the cell prepares itself for division, and during mitotic phase, actually the division takes place. And in interphase, there are again three substages: G1, S, and G2 respectively. This S phase is very crucial. It is also known as synthesis phase, and in this S phase of cell cycle, DNA replication takes place. This question is very important. It can come in the form of MCQ. It can come in the form of name the following also. So, our next question. What is nucleosome? DNA, the genetic material present inside the nucleus. And normally what happens inside our body, the DNA is approximately 2 meters in length, but the diameter of the nucleus is very short. So how this 2 meter long DNA will accommodate itself inside the small nucleus? Actually, what happens here? There is a core of 8 histone proteins. Core of 8 histone proteins. Some globular structures known as histone protein. It makes a core. And DNA winds around this core of histone protein. And this complete structure, that is histone, rather you can say core histone, along with DNA, together known as nucleosome. This nucleosome is also very important for the diagram also. Next, what are the various components of a nucleotide? First of all, you have to know what is nucleotide. Nucleotides are the unit of nucleic acid, that is DNA or RNA. You know it. So, there are three components of a nucleotide. First, one pentose sugar. Number two, phosphate group. And number three, nitrogen base. So whenever we will discussing about nucleotide, there is another term, that is nucleoside. SIDE, side. So how it looks like? It looks like this. This is the pentose sugar. In this case, it is ribose. So pentose sugar and nitrogen base. This two is known as nucleoside. And when a phosphate group comes and attach with it, it forms the complex of nucleotide, like this. So the various component of a nucleotide are a pentose sugar, a phosphate group, and a nitrogenous base. These three are the different components of the nucleotide. And whenever it is nucleoside, there will be no phosphate group. Only ribose sugar and nitrogen bases are there. Next, which bond is present between adenine and thymine and guanine and cytosine? First of all, this adenine, thymine, guanine and cytosine, these four are the nitrogenous base. Adenine and thymine pairs with each other, whereas guanine and cytosine pairs with each other. Adenine pairs with thymine by means of 2 hydrogen bond, whereas guanine pairs with cytosine by means of 3 hydrogen bond. So if the question asks like that, which bond is present in between this, the answer will be hydrogen bond. Hydrogen bond. And if the number is asked, the bond present between adenine and thymine is 2 hydrogen bond, and in between guanine and cytosine there is a 3 hydrogen bond. Next, in which stage of cell cycle chromosomes are arranged at the equatorial region? Another important question. As we know, the mitotic phase of cell division contains 4 subsequent phases: prophase, metaphase, anaphase and telophase. Among these 4 phases, metaphase is the stage where the chromosomes are arranged at the equatorial region. How? Just like this. Imagine it is an animal cell and these are the spindle fibers present in it. Chromosomes are arranged like this. This region is the equatorial region and chromosomes are very nicely arranged at that equatorial region, and whenever you will see it under a microscope, you can see the proper structure of a chromosome. So the metaphase phase is the best phase to see the proper structure of a chromosome and during the cell division period. Next, define gene. Sorry, I have missed this question. Define gene. What is gene? Genes are a specific sequence of nucleotide present on a chromosome, and that will represent a particular character. Again, I am saying genes are the specific sequence of nucleotides arranged on a chromosome. Not only that, which will determine a particular character. Just like this. Stretch of nucleotide. This small segment, that is stretch of nucleotide, you can consider as a gene. And this gene is responsible for expressing a particular protein. For example, you can say the height or hair color, whatever. So, specific sequence of nucleotide arranged on a chromosome which will determine a particular character. Next question. Prophase, metaphase, anaphase, telophase characters. These 4 are the subsequent phases during the karyokinesis or the mitotic division. There is some specific character of each phases, like prophase. In this phase, what happens? Initially, during interphase, the chromosomes are in the form of chromatin fiber. During prophase, the chromatin fiber absorbs water from the surrounding, from the nucleoplasm, and it becomes short, thick, and forms the structure of chromosome. Spindle fiber starts to appear. Centriole moves to the opposite pole. So these are some specific features during the prophase. Second, metaphase. Metaphase is the phase of cell division where the chromosomes are arranged at the equatorial pole. Anaphase. During anaphase, the chromosomes are separating towards each pole. And the last one is telophase. In this phase, the individual chromosomes separated on the opposite pole again will make a new type of cell. The nuclear membrane and nucleolus will reappear, and finally the cytokinesis will take place after telophase. So in your book there are some specific characters given for each and individual phases of cell cycle. For each stages, at least remember two specific character with diagram. Very, very important. Next, mitosis versus meiosis. There are two types of cell division in our body. First one is mitosis, second one is meiosis. What happens during mitosis cell division? The mother cell will give rise to daughter cell. Mitosis. The mother cell diploid will give rise to daughter cell having equal number of chromosome. So you can see here that from a mother cell two daughter cells are produced. These two are the daughter cells, and this one is a mother cell. So one mother cell will give rise to two daughter cells during mitosis. The number of chromosome in mother cell and daughter cells are equal. And this mitosis side of cell division we can find in our somatic cell, that is a body cell. That is the cells of liver, pancreas, kidney, skin. All the cells are divided by means of mitotic cell division or mitosis. The second type of cell division is meiosis. What happens here? The mother cell initially is diploid. It will give rise to two daughter cell having half of the number of chromosome compared to mother cell. This phase is meiosis 1. Meiosis 1. After that, these two daughter cell eventually will divide again. So we can see here at the end during meiosis cell division, from one mother cell, four daughter cells are produced. But in mitosis, from one mother cell, two daughter cells are produced. In mitosis, the number of chromosomes in mother cell and daughter cells are equal. In meiosis, the number of chromosomes in mother cell and daughter cells are not equal. In the daughter cell, the chromosomes are becoming half. And that is why another important question from this part, meiosis cell division is known as reductional division. Give reason can come, like why meiosis cell division is considered as reductional division, because the number of chromosome in mother cell becomes half within the daughter cell. The chromosome number is being reduced here, and that's why it is considered as reductional division, whereas mitosis is considered as equal division, because the number of chromosome is remaining same in the mother cell as well as in the daughter cell. Okay. So another thing, in which type of cell we will find meiosis? Basically gamete cell. Gamete cell in the sense testis, ovary. in case of animal, and in case of plant you can say pollen and egg. This type of cell divide by means of meiosis cell division. So summarization of these differences: mitosis cell division is known as equal division, meiosis is known as reductional division, because here the number of chromosomes are remaining same, here the number of chromosomes are becoming half. Second, from during mitosis cell division, from one mother cell, two daughter cells are produced. Here from one mother cell, four daughter cells are produced. Third, mitosis cell division we can find in the somatic cell. That is the cells of skin, liver, pancreas, kidney. But in case of meiosis cell division, we will find in the gamete cell. Testis and ovary in case of animal, and pollen and egg in case of plant. Last question, karyokinesis versus cytokinesis. Karyokinesis, the word karyon, it means nucleus. So the division of nucleus is known as karyokinesis, whereas the division of cytoplasm is known as cytokinesis, because during cell division first the nuclear division takes place, that is karyokinesis will take place. After that the cytokinesis will take place and the cell will become into two half. So karyokinesis is the division of nucleus and the cytokinesis is the division of cytoplasm. So this is all for this chapter, chapter number 1, structure of chromosome, cell cycle and cell division. Okay. Now I am moving to the next chapter. Chapter number 2, genetics. Very important and scoring chapter. First of all, some numerical part you can get from this chapter, like solving of the cross, the ratios of the monohybrid and dihybrid crosses, like this. So first question from this chapter, define allele. So what is allele and what is the difference between a gene and allele? Basically, alleles are the alternative forms of a gene. For example, I am taking one example like the gene of height. The gene of height. So height normally can be out of two types. Either it can be tall or it can be short. Either or. These tall and short are the allele. So height is a gene and tall and short, these are the alternative. Either the height can be tall or it can be short. So the alternative form of a gene is known as allele. Skin color, dark or fair, allele. And one gene will contain minimum 2 allele and maximum multiple. Okay. So define allele, it is the alternative form of a gene. Next, what are autosomes and allosomes? Another important question. Inside our body cell rather, there are two types of chromosomes. Some chromosomes which are responsible for carrying the somatic character, that how our height will be, how our skin color will be, how our hair texture will be. So the somatic character bearing chromosomes are known as autosomes, whereas allosomes are the chromosome that are identification of our sex. That means based on allosome, it is determined whether an individual is a male or a female. I mean to say, autosome are the somatic chromosome, whereas allosomes are the sex chromosome. And allosomes are of two types, X or Y. How many autosomes are present inside a cell? 22 pair. And the number of allosomes is one pair. So here 44 plus 2 is equals to 46 chromosome in a single cell. So autosomes are the somatic chromosome which will determine the somatic characters of our body. On the other hand, allosomes are the sex chromosome which will determine the sex of an individual. If it contains two X chromosome, then the individual is a female. If it contains one X and one Y chromosome, the individual is a male. Next, example of two X-linked inherited disease. X-linked inherited disease means the diseased genes are present on the X chromosome. And the examples like hemophilia, color blindness, these two are the example of X-linked inheritance disease. Hemophilia and color blindness. Color blindness, the affected individual cannot differentiate between red and green. This is color blindness. And hemophilia, the blood clotting will not take place. That means the bleeding will not stop. If any injury will take place, it will continue. Continuously bleeding will take place and it can be fatal also. So hemophilia and color blindness, these two are the example of X-linked inherited disease. Next question. Monohybrid cross, phenotypic and genotypic ratio. Monohybrid cross means the cross between two individuals considering one pair of, monohybrid cross. Monohybrid cross are the cross between two individuals considering one particular character. For example, a tall pea plant is crossed with a short pea plant. So here we are considering one character, that is the height, and one character means two allele. So the phenotypic ratio of the monohybrid cross is 3:1 and it is constant, and the genotypic ratio is 1:2:1. It is also constant. Next, solve of monohybrid and dihybrid cross with checkerboard. Section B, 5 marks questions. It can come from this part. A condition will be provided on the question paper and you have to solve this by using monohybrid or dihybrid cross given on the question paper. So this is very important. It can come as a whole in section B. Next, genotypic ratio of dihybrid cross, and the genotypic ratio of dihybrid cross is 9:3:3:1. Next, law of independent assortment. Generally what happens, this law of independent assortment and the solve of the cross comes together as a unit. Anyways, law of independent assortment. This is the law of genetics given by Mendel. What does it implies? As we know, assortment. For example, it is a condition of a seed of pea plant having one pair of character. For this part, it's representing the height, and this part is representing the seed shape. The height can be either tall, denoted by capital T, or it can be short, denoted by small t, whereas the seed shape R, capital R denotes round and small r denotes wrinkled. So it's a heterozygous condition. So when gamete will form, we know that allele will be segregated. Now, From this pair, one pair of allele is there for this character, one pair of allele is there for the second character. From this pair, either capital T comes in the gamete, and from this pair, just think capital R is there. Another combination can be like this: from this pair capital T, from this pair small r, from this pair small t, from this pair capital R, from this pair capital T, from this pair small r. No, it's a given. Small t, small r. See, there are 4 possible combinations of gamete, and from this pair, the allele which comes is completely independent upon the allele which will come from this pair. For example, from here capital T can come, from here capital R can come, or from here capital T, small r, small t, capital R, small t, small r. There are variable combinations. Okay? Independent assortment. Assortment indicates segregation, and the segregation of this allele is completely independent for the segregation of second pair of allele. Anything can come to make a pair, but not like this capital T and small t will come together. No, because these two are the allele for a particular same gene. Again, capital R and small r, these two will not come together. It's a random combination, and this law is known as independent assortment, and this is only applicable, exclusively this is applicable for a dihybrid cross. Okay. What are homologous chromosomes? Homologous chromosomes are chromosomes which are structurally similar in shape, in size, and each obtaining from an individual parent. That means one chromosome will come from father, another chromosome will come from mother. These two chromosomes are structurally similar, similar in shape, similar in size, and the position of the genes on the chromosomes are also similar. And these two chromosomes are known as homologous chromosome, and these two will make a pair in our cell. So homologous chromosomes are structurally similar chromosome, each obtaining from individual parent. Next, why Mendel has selected the pea plant for his experiment, and what is the scientific name of pea plant? The scientific name of pea plant is Pisum sativum. Whenever you are writing a scientific name, just make it sure the underline should be there individually. The first letter of the genus should be capital always, and the first letter of the species should be small always. Otherwise, you will not get marks. The scientific name of the pea plant is Pisum sativum. And why Mendel has selected pea plant? For several reasons, Mendel has selected pea plant. The first one is pea plant is a bisexual plant. That means it have both the male and female reproductive organ. Second, very short lifespan. Third, contains 7 specific character. 7 character means 14 alleles are there. This is an important reason. Acha. Next, faster growth rate. So these are the probable reason for this. Mendel has selected pea plant for performing the experiment of monohybrid and dihybrid cross. So, the second chapter genetics is over. Now, chapter number 3, absorption by root. The first question, which is very common from this chapter, that is, name the process by which water enters into root hair. That is endoosmosis. If you write osmosis, you will get half marks because there are two type of osmosis. From the first, in the first type, endoosmosis, the water will enter into the root hair, and in exoosmosis, water will leave the root hair. So the answer will be endoosmosis. Next, difference between diffusion and osmosis. Diffusion and osmosis both are the example of passive transport. But in case of diffusion, both solute and solvent can pass from the region of their lower concentration to the extremely sorry. The second question, difference between diffusion and osmosis. Diffusion and osmosis, these two are the passive transport inside our cell. During diffusion, both the solute and solvent, both the solute and solvent can pass from the region of their higher concentration to the region of their lower concentration. While in case of osmosis, only the solvent molecule can pass from the region of their higher concentration to the region of their lower concentration. This is the first difference between diffusion and osmosis. And the second one is, in diffusion, there is no separating medium between the two medium. For example, from this side to this side, the transportation is taking place. So there is no medium, no barrier in between the two cells in diffusion. But in osmosis, there is a semi-permeable membrane in between the two medium, and which will allow selective materials to pass through it. Selective material in the sense, but I have said that during osmosis, only water molecule or solvent molecule will pass. No solute can be passed from here. So, the two difference between diffusion and osmosis. Next, in which type of transport ATP is required? Answer is active transport. So, why ATP is required here? During diffusion and osmosis, in both the cases, the molecules are passing from the region of their higher concentration to the region of their lower concentration. But in this third type of transport, that is active transport, here the molecules or ions, specifically ions, are passing from the region of their lower concentration to the region of their higher concentration. That is against the concentration medium, from the region of their lower concentration to the region of the higher concentration. The ions are passing. That is why externally ATP is required. Next, what is ascent of sap? As we know that the root hairs are present inside the soil, are responsible for absorption of water. And after that, the water will reach to the surface of the leaf by means of xylem vessel. And this movement of water molecule through the xylem vessel is known as ascent of sap. Next, define transpiration pool. What is a transpiration pool? For this question, first you have to know what is transpiration. Transpiration is the release of the excess water from the aerial part of the plant in the form of water vapor. Suppose these are the root hairs and that are responsible for absorbing water molecules from the soil. This water molecule will go upward by the help of xylem vessel and eventually reaches to the surface of the leaf. Within the leaf, the preparation of food takes place, that is photosynthesis will take place, and after that the excess water will release from the surface of the leaf in the form of water vapor. So if the water vapor will release, that is transpiration will take place, this leaf surface will become empty, and for this reason the transpiration will exert a pull or force so that more water can be absorbed by the root hairs. So the transpiration will create a pull and the water molecule, more water molecule will go upward by means of xylem to fill up this space. And this pull or force, you can say, generated by transpiration is known as transpiration pull. Okay. So now the next question is, if you sprinkle some common salt on grass growing on a lawn, it is killed at that spot. Give reason. This is another important question from this chapter. So basically what happens? When we place, wait, let me clean this. When we place some common salt over the grass cells, the surrounding medium of grass cells contains high concentration of salt. The surrounding medium of grass cell contains high concentration of salts, and we can say also low concentration of water compared to the inside of the grass cell. And inside the grass cell, the water concentration is high and salt concentration is very low. And as we know, osmosis takes place from the region of their higher concentration to the region of their lower concentration. So in this case, what will happen? Inside the grass cell, the water is higher, and outside the grass cell, the water is lower and salt is high. So water will come out from the cell. Water will come out from the cell. That means you can say plasmolysis will take place and the grass cell will die. This is the theory. So whenever we will sprinkle some common salt over some grass cell, it makes the surrounding medium of grass cell hypertonic, and in an hypertonic solution plasmolysis will take place, that the cell will burst out and eventually the cell will die. Next, a plant cell, when kept in hypertonic solution for 30 minutes, it turns flaccid. Give reason. Same theory, just application is different. A plant cell, when kept in hypertonic solution for 30 minutes, it turns flaccid. Give reason. Just remember one thing, it's very simple. What is the basic difference between a hypertonic and hypotonic solution? For example, here I am taking an hypertonic solution, and on this side, hypotonic solution. So in a solution, as we know, there are two parts. One is solute, another one is solvent. So in case of a hypertonic solution, the solute is high and the solvent is low. Solvent means, in this case, you can take example as water. So in case of hypertonic solution, solute is more and solvent is low, where outside the cell. Hyper means more. So outside the cell, if the solute concentration is higher, then we can say that it's a hypertonic solution. Whereas in case of hypotonic solution, the solute is low, hypo means less or low, and solvent, that is water, is high, where outside the cell. So when we will place any cell into a hypertonic solution, just take an example. This is an hypertonic solution in which we have placed a cell. So in hypertonic solution, outside solute is higher and water is lower, and inside water is higher, solute is lower. Water will move from higher concentration to lower concentration like this. The cell will perform plasmolysis, or you can say the cell will shrink and eventually dies. But when we place some cell in a hypotonic solution, the opposite phenomenon will take place. Hypotonic solution means outside the water is higher, inside water is lower. So water will come inside. In the surrounding medium, the water will come inside. The cell will swells up. We can say deplasmolysis will take place. So what was our question? A plant cell, when kept in hypertonic solution for 30 minutes, it turns flaccid. It turns flaccid means it will give out the water molecule. The cell will give out the water molecule. The water will move this direction. So this is all the question, all the important question which can come from this chapter, absorption by roots. Our next chapter is transpiration. The first thing we will discuss from this chapter, that is, what is the difference between transpiration and guttation? Transpiration is the release of water molecule through the surface of the leaf in the form of water vapor. As we know, the root hairs are responsible for water absorption from the soil, and water will goes up by means of xylem vessel and eventually reaches to the surface of the leaf. Water will reach eventually to the surface of the leaf. Here the water will utilize for the purpose of photosynthesis. It will make food molecules, and after that the excess water molecule will evaporate in the form of water vapor. And this evaporation of excess water from the leaf surface, or you can say from the aerial parts of the plant, in the form of water vapor is known as transpiration. And in case of transpiration, the water vapor is releasing through the opening known as stomata, present on the epidermis of the leaf. What about in case of guttation? The excess water will come out in the form of droplet. In the form of water droplet. And there is another opening known as hydatodes. Where it is located? It is located in the margin of the leaf. For example, this is a grass type of leaf. Its margin contains a single opening. This type of opening. These are known as hydatodes. And from hydatodes, the water molecule will release drop by drop in the form of droplet, and this is known as guttation. Next, name the opening through which guttation takes place. Answer is hydatodes. Next question, structure of stomata with diagram. Stomata are the minute opening present on the surface of the leaf, or you can say the surface of the epidermis. These two are the guard cell. And on the outer surface of the guard cell, there are some epidermal cells or subsidiary cells are present, which will provide water molecule. And this opening guarded by guard cell is known as stomata. These are the epidermal cells. When the guard cell becomes turgid by absorbing water molecule from the surrounding cell, when the guard cell becomes turgid by absorbing water molecule from the surrounding cell, the stomata remains open. And when the guard cell will lose water, the guard cell will become flaccid and the stomata will be closed. So, guard cell turgid, it means open stomata, while guard cell flaccid, it means closed stomata. And from where the guard cell will get water to become turgid or to become flaccid? From the epidermal cells surrounding the guard cell. Next, what are lenticels and cuticle? Lenticels are the smaller opening, very minute opening present on the surface of the stem. As we can say, that is a bark. The woody stem contains a cover layer surrounding it known as bark. And on the surface of the bark, there are some small openings known as lenticel. And through this lenticel, a little bit amount of transpiration will take place. We can say that the majority of the transpiration, that is 90% of the transpiration takes place by stomata, and the remaining 10% takes place by lenticel, the small opening present on the bark, and the third one is cuticle. What is the cuticle? This is the waxy layer present on the surface of the leaf. The surface of the leaf contains an waxy layer that is known as cuticle. So, a very little bit amount of transpiration will take place by means of lenticels and cuticle. Next, various factors which controls the rate of transpiration. There are some internal and external function which regulates the rate of transpiration. For example, if the intensity of sunlight, factors regulating transpiration. If the intensity of sunlight is high, the transpiration will be more. Why? Sunlight intensity is high, it will regulate the absorption of water more, and if the absorption water becomes more, the rate of transpiration will be as well as increases. Next, the factor is wind velocity. This wind velocity is directly proportional to the rate of transpiration. The more wind velocity, it indicates the more rate of transpiration, but after a certain time, if the wind velocity becomes much higher than the normal, then the rate of transpiration will fall down. So initially wind velocity will promote the rate of transpiration, but after crossing a saturation level, when the wind velocity is increasing and increasing, the rate of transpiration will be lower. The third factor, atmospheric pressure. Which also regulates the rate of transpiration. Another factor is, you can say, carbon dioxide, sorry. humidity. If the air contains excess amount of water vapor in it, the air is already wet, then the rate of transpiration will be lower in that case. So what you will do, you will just remember these different factors of transpiration which are directly proportional to the rate or which are responsible for declining the rate of transpiration. For example, sunlight, wind velocity, atmospheric pressure, these are increasing the rate of transpiration. On the other hand, humidity, surface of the leaf, these are responsible for declining the rate of transpiration. So these factors regulating transpiration are very important. Next, example of a plant with sunken stomata. Answer is nerium. Nerium leaf contains stomata which are very sunken. The opening is very small. So the amount of water releasing through the transpiration mechanism is very less. Adaptation of plant for transpiration. There are several adaptation in plant which reduces the rate of transpiration. What are these? The leaf will contain a greater level of cuticle, and cuticle will provide obstructions during the release of water through transpiration. So presence of cuticle, sunken stomata, presence of hair on the stomata, these are considered as the adaptation for transpiration in plant. Next question. Forests tend to bring rain. Justify the statement. In a forest area, the number of aerial part or number of trees are much higher. So this tree will release a lots of water molecules by transpiration mechanism, and this water molecule will goes up, forms the cloud, and finally this cloud are responsible for bringing rain. So this is another important explanation or important question you can say in the segment of give reason from this chapter. Next, some plants show wilting of their leaves at noon even when the soil is well watered. Give reason. Very important question. Some plants show wilting. Wilting means flaccidity of their leaves at noon even when the soil is well watered. During afternoon, the rate of absorption is lesser than the rate of transpiration. the rate of absorption is less than the rate of transpiration during afternoon. So, the amount of water plant absorbs, for example, 1 ton, is much lesser the amount of water plant releases. Plant releases more, for example. That is why the leaves become wilted, even the soil is well watered. The rate of absorption is much lesser compared to the rate of transpiration. Last question. Oil is Why oil is placed on the surface of water during the experiment of transpiration? Which instrument measures the rate of transpiration? First, I will answer the second part of this question. Which instrument measures the rate of transpiration? The answer is potometer. There are 4 types of potometer. One is Gannon's potometer. Second one is Farmer's potometer. Third one is Darwin's potometer. And fourth one is Gaidiou's potometer. So all the type of potometer, especially the Gannon's potometer, are used to measure the rate of transpiration. And why oil is placed on the surface of water? Oil is placed on the surface of water to inhibit evaporation during the experiment for establishing the transpiration. So oil is placed on the surface of water to prevent or to inhibit evaporation from the water surface. So these all are the important questions from this chapter, transpiration. Our next chapter is photosynthesis. First question, structure of chlorophyll and chloroplast. Chloroplast is a type of plastid which contains the green pigment chlorophyll. So, what are the various parts of a chloroplast? A chloroplast contains, it's a double membrane wound structure as we know, present only in plant. It contains several stack-like structure, stack-like coins placed one after another, known as grana. And these are also interconnected. The stack of coins are known as grana. In singular, it is known as granum. And in plural, all the granas together known as grana. And the matrix on which the grana is present, this matrix, is known as stroma. Inside the grana, for example, each stack is known as thylakoid. And inside the lumen of thylakoid, the pigment chlorophyll is present. So this is the structure of chloroplast. Now the structure of chlorophyll, it contains several elements like carbon is there, hydrogen is there, oxygen is there, nitrogen is there, magnesium is there. These are the elements which construct the molecule of chlorophyll. Carbon, hydrogen, oxygen, nitrogen and magnesium. It's a green pigment, but it is present inside the lumen and membrane of thylakoid. Next question, equation and definition of photosynthesis. The definition of photosynthesis is very simple. It's a physiological process in plant in which the plant makes their own food by acquiring the inorganic material from the environment like carbon dioxide, water, sunlight and using chlorophyll, and as a result they produce starch or you can say glucose. Along with that, oxygen gas is produced, which is liberated in the atmosphere, and as well as some amount of water is also generated that are reused in the process of photosynthesis. And what is the equation of photosynthesis? The reactants are the glucose, oxygen. Here you will write sunlight and chlorophyll. So carbon dioxide, water, by using sunlight and chlorophyll, they will produce glucose, oxygen, and water molecule. This is the complete balance equation of photosynthesis. Next question. Define photolysis. This photolysis is also known as Hill's reaction. As per the name of Robert Hill, who discovered first this phenomenon. Robert Hill. So what is photolysis? During photosynthesis, first the chlorophyll remains inactive inside the leaf. Inactive chlorophyll. When the smallest particle of sunlight, that is photon, falls on it, the chlorophyll becomes activated. Initially the chlorophyll was inactive. When the smallest particle of sunlight, that is photon, falls on it, it becomes activated. Now this activated chlorophyll will break down water molecule, and as a result water molecule will split up into H plus and OH minus ion. So this lysis or breakdown of water molecule by activated chlorophyll and photon or sunlight is known as photolysis. Photo means light, lysis means breakdown. By using light energy, by using solar energy, we are breaking down water molecule and generating H plus and OH minus ion. And this event is known as photolysis or Hill's reaction according to the name of Robert Hill. We can jot down the equation of photolysis. This is the equation of photolysis. If the definition comes, try to give the equation along with the definition. Photolysis, breaking down of water molecule by using activated chlorophyll and produces 4H plus and OH minus radical. Next, the full form of NADP. Full form of NADP is nicotinamide adenine dinucleotide phosphate. This is the full form of NADP, nicotinamide adenine dinucleotide phosphate. Next, what is polymerization? Polymerization is a process by which glucose molecules are converted into starch. Generally, during photosynthesis, plant produces glucose molecule at first. Now this glucose molecule will polymerize, will join by a bond, will make a polymer to form starch. So this process is known as polymerization. Next, what are the end product of light phase and dark phase? So the end product of the light dependent reaction or light phase are number one is NADPH, number two is ATP, number three is oxygen, number four is water. These four are the end product of light dependent phase or light phase, whereas the end product of dark phase, this is glucose. Along with that, NADP, ADP, etc. So end product of light phase or light dependent phase and the end product of dark phase or light independent phase. Last question from this chapter. Sorry, second last. Factors controlling the rate of photosynthesis. It's like transpiration. There are several factors are there like sunlight, concentration of carbon dioxide, presence of water molecule, presence of chlorophyll. These all are related to the rate of photosynthesis. For example, if the concentration of the carbon dioxide high, it will prefer the rate of photosynthesis because during photosynthesis CO2 is needed as a reactant. So if the CO2 concentration is high, it will increase the rate of photosynthesis. If the sunlight is also high, it will increase the rate of photosynthesis after a certain limit. Along with that, water molecule and chlorophyll, these two are also playing an important role by increasing the rate of photosynthesis. The last question from this chapter is iodine test. We use this iodine test to perform the presence of starch in a leaf. So inside a leaf, the starch is present or not, it is proved by the iodine test. How? If in a experiment of photosynthesis, we will pluck one leaf to establish that it contains starch or not, we will just pour some amount of iodine solution on the surface of the leaf. If it turns blue-black, iodine solution. If the leaf turns blue-black, it indicates that starch is present there. In another set of experiment, if we give iodine solution, same, but the leaf turns brown, not blue-black, it indicates that starch is absent. So by using iodine test, we can conclude a leaf contains starch or not. So these all are the important questions from this chapter, photosynthesis. The next chapter is chemical coordination in plants. The first question, which is very important from this chapter, that is name the plant hormone which is gaseous at room temperature. All the plant hormones, for example, auxin, gibberellin, cytokinin, abscisic acid, these all are liquid at room temperature, except the one that is ethylene. This is the only hormone which is gaseous at room temperature. Along with that, you will just find out the function of ethylene, that it helps in the process of fruit ripening. Next, which plant hormone helps in phototropism? Auxin. Auxin is a plant hormone which helps in the process of phototropism. Next question, ABA and IAA full form. Full form of ABA is abscisic acid. And full form of IAA is indole 3 acetic acid. This IAA is a synthetic form of auxin. Synthetic auxin. The full form is indole 3 acetic acid. Next, example of thigmotropism. There are two types of plants, for example, cuscuta, grapevines. These plants are performing the mechanism of thigmotropism, where the stimulus acts is touch. Touch is the stimulus. And by receiving the stimulus, the tendrils of the plant, the tendrils of the green plant, pea, cuscuta, grapevines, they will just climb up around a hard surface. Next, positive phototropic and positive hydrotropic plant part. Phototropic plant part means the part of plant which moves towards the sunlight. And as we know, if a seed is implanted, the shoot part will grow towards the sunlight and the root will move towards the center of gravity. So the shoot is a positive phototropic as it is moving towards the sunlight, whereas the root is the negative phototropic. It is going away from the sunlight. On the other hand, root is the positive geotropic because it is moving towards the center of gravity, and shoot is the negative geotropic. It is going away from the center of gravity. So these examples are very important: positive phototropic, positive hydrotropic. Even positive hydrotropic is also root because root moves towards the source of the water present under the soil. So these examples are very important, and mainly from this chapter, so short question will come. From this thigmotropism, one long question you can expect by giving a pictorial question. It may come thigmotropism, but other question will come in the section A. Okay. Our next important chapter is circulatory system. First, name the metal present in hemoglobin. Iron. Hemoglobin is a iron containing metal, and the color of iron is reddish brown, which imparts the red color of hemoglobin as well as the red color of blood. So hemoglobin is a metal, iron is a metal present in hemoglobin. Next, which metallic ion and vitamin helps in blood clotting? The metallic ion helps in blood clotting is calcium, and the vitamin which helps in is vitamin K. Basically, vitamin K helps in the formation of fibrinogen. And as we know, fibrinogen is a very important plasma protein which takes part effectively in the process of blood clotting. So calcium is a metallic ion which helps in the process of blood clotting, and vitamin K is a vitamin which helps in the formation of fibrinogen as well as promotes blood clotting mechanism. Next question, name the thinnest blood vessel. There are three types of blood vessels present in our body: artery, veins and capillaries. Among these three, the thinnest blood vessels is capillary because it has no muscular wall. Only epithelial cells are there, endothelial cells lining the lumen. This is the lumen through which the blood will pass, lumen, and these are the endothelial cell layer. So capillaries are the thinnest blood vessel. Next, difference between WBC and RBC. So first we will discuss some structural difference, then we will go for functional difference. The RBC or red blood cell, another name of this RBC is erythrocyte, whereas the another name of WBC is leukocyte. Next, the RBC is a biconcave shape. No nucleus is present. And WBC is amoeboid shape. It does not have a specific shape, amoebic shape, and nucleus present. Next, RBC contains hemoglobin, the red pigment hemoglobin RBC contains, and WBC is colorless. It does not contain any pigment. Next, the lifespan of RBC is 120 days, and the lifespan of WBC is 14 to 20 days on an average. Next, RBC produces inside red bone marrow, and WBCs are produces inside red as well as yellow bone marrow. So these are the basically structural difference of RBC. Now the functional aspect. RBC helps in the transport of oxygen and carbon dioxide throughout our body, whereas WBC helps in the process of immunity provide, inflammation, pus formation, phagocytosis, allergy reaction, etc. There are different type of WBCs are present in our body. For example, neutrophil, eosinophil, basophil, monocyte, lymphocyte, and each kind of WBC performs a different type of activity. Like lymphocytes are responsible for immunity production by producing antibodies. Monocytes and neutrophils are responsible for phagocytosis. Eosinophils are responsible for allergic reaction, and basophil produces heparin, which is the natural anticoagulant. So these are the also very important part which is given on a book in your book on in the form of a chart. So different type of WBC and their respective function. This segment is also very important. Next, yeah, types of WBC with its function. 5 type of WBCs are there. As I said, monocyte, lymphocyte, neutrophil, eosinophil, basophil. And these 5 type of WBCs have different type of functions. So these are very important. Universal donor and universal receiver blood group. Universal donor blood group means it can give blood to anyone. That is O positive. And universal receiver means it can take blood from anyone. That is AB positive. Next, name the covering of the heart. The covering of the heart is pericardium. The covering of the heart is pericardium, and this pericardium also has 2 layers. The outer layer of pericardium is parietal and the inner layer is visceral pericardium. And in between these two layers, lubricant fluid is present known as pericardial fluid. So pericardium is the name of the covering of heart. It is a bilayered sac. The outermost layer is parietal pericardium, the innermost layer is visceral pericardium, and in between these two layers, a lubricant fluid is present known as pericardial fluid, which inhibits the friction during the cardiac activity. Next, natural pacemaker of heart. The function of pacemaker is to generate the impulses for contraction and relaxation of the heart, and the natural pacemaker of heart is SA node, and the full form is sinoatricular node, SA node or sinoatricular node. This is the natural pacemaker of heart. Next, the difference between artery and vein. Another important question. Here also first we will discuss the structural aspect, then we will discuss the functional aspect. So let me draw a diagram. Here you can see the layers of artery, muscular layer. The outermost layer is tunica intima. Middle layer is tunica media. And the inner layer is tunica adventitia. But here clearly you can see the layers of muscular wall is much thicker compared to the vein. So if the layers of the muscular wall is thicker, the lumen will be narrower, which is in case of artery. But in case of the vein, the layers are thinner comparatively. So and the lumen is wide. So in case of artery, the muscular wall is thick, the lumen is narrow. In case of vein, the muscular walls are thin and the lumen is wider. Next difference you can say, there are no valve present in artery. No valve is present. And in case of vein, valves are present. Next, the functional aspect. The artery carries the pure blood, or you can say oxygenated blood. Oxygenated blood means more oxygen, less carbon dioxide. So the artery carries the oxygenated blood or pure blood from heart to all over the body. Whereas a vein carries the impure blood or deoxygenated blood, that means more carbon dioxide, less oxygen. From the different part of the body, the vein collects a deoxygenated blood and brings to the heart. So the artery carries oxygenated blood and the vein carries deoxygenated blood. There is one exception, pulmonary artery and pulmonary vein. Pulmonary artery is a specific type of artery which carries impure blood or deoxygenated blood. And pulmonary vein, specific type of vein which carries oxygenated blood, that is pure blood. These two are the exceptions. All the arteries carry pure blood except pulmonary artery. All the vein carries impure blood except pulmonary vein. Next, double circulation through human heart. Pictorial question can come from this part, double circulation. Why it is known as double circulation? Because two type of circulations are taking place inside our heart. One is systemic, another one is pulmonary. So let me describe this. It's an random drawing. This is a very basic heart diagram, like this. So double circulation. It is consisting of two type of circulation. One is systemic, another one is pulmonary. What happened first? The impure blood or deoxygenated blood from the upper and lower part of the body comes into the heart by means of superior vena cava and inferior vena cava. This is our SVC, superior vena cava. This is IVC, inferior vena cava. So impure blood or deoxygenated blood collecting from the whole body are coming inside the right auricle. This is the right auricle, right ventricle, left auricle, left ventricle. So comes inside the right auricle. From right auricle, this impure blood goes down to the right ventricle. From right ventricle, it will enter into the pulmonary artery. As I have said, pulmonary artery is the exception which carries impure blood. Finally, pulmonary artery enters into respective lungs, and this impure blood reaches to the lung for the purpose of the blood filtration. Gaseous exchange will take place. The blood will become oxygenated. After oxygenation of the blood, from the lungs, the blood will enter into the left auricle of the blood, heart chamber. Through pulmonary veins. The pure blood will come in the chamber of left auricle. From the left auricle, the blood will goes down to the left ventricle, and from left ventricle, it will enter into the aorta that will distribute the pure blood or oxygenated blood to all over the body surface. So here the various parts of the heart chambers are involved. For example, body, SVC plus IVC, right auricle, right ventricle, lungs, pulmonary artery, lungs, pulmonary vein, left auricle, left ventricle, dorsal aorta, again goes to the body. So this is the pathway of circulation in which from this part, right ventricle to left auricle, this much pathway is the pulmonary circulation, where the lungs are involved, and the remaining part, that is from left ventricle up to right auricle, where the body part is involved, is the systemic circulation. So body involves systemic circulation, lungs involve pulmonary circulation. Together these two are known as double circulation in human heart. This is also an important question. Pictorial questions can come from here. With this question, this chapter is covered, circulatory system. Our next chapter is excretory system. First question, what is Malpighian corpuscle? As we know, the unit of the main excretory organ kidney is nephron. Now each nephron has two parts. One is Malpighian corpuscle. Second one is renal tubule, which again divided into PCT, loop of Henle, DCT, and collecting duct. This Malpighian capsule or Malpighian tubule again is divided into two parts. One is Bowman's capsule, another one is glomerulus. Together these two known as Malpighian corpuscle. The diagram is like that. It's a cup-shaped structure, Bowman's capsule, on which a tuft of blood capillaries are present, fits inside the Bowman's capsule, are known as glomerulus. So this is this is glomerulus. This part is glomerulus. And this is Bowman's capsule. Together these two is known as Malpighian corpuscle. And this is the afferent arteriole. And this is the efferent arteriole. Next, name the pigment present in urine. This is urochrome. Urine contains a pigment urochrome which imparts a yellowish color of urine. Next, steps of urine formation. There are three sequential steps of urine formation. Number one is ultrafiltration. Number two is reabsorption, rather selective reabsorption. And the third one is tubular secretion. Next, what is vasa recta? Vasa recta is a network, tubes of network, which arises from the efferent arteriole and spreads all over the renal tubular part of nephron and eventually connects with the vein, renal vein. So what is the function of vasa recta? As we know that when the blood gets filtered, the afferent arteriole brings lots of water along with the afferent arteriole it brings a lots of water, solutes and bloods. Where? Inside the glomerulus. So glomerulus is a tuft of blood capillary which will sieve this element and it will allow to pass the filter medium, the liquid medium through the renal tubule, and the large particles like protein, blood cells and the important material are again goes back through efferent arteriole. Where? Through efferent arteriole it will enter into vasa recta, another network tube, and vasa recta will eventually return this to the renal vein. hematuria and glycosuria. Hematuria is the presence of blood in urine, and glycosuria is the presence of glucose or sugar in urine. Next question, role of ADH in urine formation. ADH is an important hormone known as vasopressin or antidiuretic hormone. This ADH or vasopressin helps in the absorption of water molecule during urine formation. Why? Because all we need are producing a more concentrated urine on which the water should be less. And ADH hormone helps in the process of water reabsorption. So if the ADH hormone is not present in our body, then the water will be present in the urine. The urine will be more dilute and it will lead to dehydration or diuresis. ADH hormone is very important hormone. It is secreted from the posterior pituitary gland. Posterior pituitary gland. Next, dialysis. Dialysis is the artificial kidney, and the patient whose kidneys are non-functional, one kidney or both the kidneys are non-functional, the dialysis is a mechanism through which the blood is passing through the dialyzer, where the blood is filtered, the urea and the other excretory materials are getting filtered, and the pure blood is entered into the human body. So dialysis is a process of artificial excretion. Name the structural and functional unit of kidney. This is nephron. Next, accessory excretory organ. Examples are skin, which is responsible for releasing sweat, a little bit of water and salt. Liver, which detoxifies any kind of drug, metals and poisons. Lungs, which releases carbon dioxide in the form of excretory gas. So skin, liver and lungs, these three are the accessory excretory organ, and the main excretory organ is kidney. What is micturition? Micturition is a release of urine upon receiving an impulse from our brain. When the bladder gets squeezed, the urine comes out from our body. That process is known as micturition. And last question from this chapter, we urinate fewer times in summer than winter. Justify the statement. This is another very important question which can come from this segment. We urinate fewer times in summer compared to winter. Why? Because during summer time, we can release water in various different ways. A little bit amount of water releases by the secretion of sweat from our skin. A little bit amount of water vapor releases through our warm air which we exhale. But in case of winter, the sweating is very less, very minute. So the most water present in our body, excretory mechanism, the water is releasing by means of urine only. There is no other alternative. So in case of winter, the urine is much dilute and we urinate frequently. But in case of summer, we urinate fewer times and the urine is more concentrated. More concentrated urine. So it was the last question from this chapter, excretory system. Next. The nervous system. Another important chapter. First question is, define synapse. Synapse is a junction between two adjacent neurons. Just like this. This is one synaptic knob. This is another. The junction between these two is known as synapse. Next, what is reflex action and reflex arc? First, let me draw the word chart. This is the path of reflex arc. First, what is reflex action? It's a spontaneous action, sudden action, and automatic action generated inside our body upon receiving a stimulus for providing a response. For example, if by mistake we touch a hot object, a hot coffee mug or soup bowl, so instantly we will remove our hand from that place. This is an example of reflex action. So when we are touching an hot object, the receptors present in our skin will receive that stimulus. The stimulus will send to our brain by means of sensory nerve. Sensory nerve will carry the stimulus to our brain and spinal cord or spinal cord. From brain and spinal cord, the command will come by means of motor neuron. Where it will come? In the muscles or the gland. And then we will just remove our hand. So this spontaneous, automatic, sudden reaction after receiving a particular stimulus is known as reflex action. And for this mechanism, the pathway which is followed in our body is known as reflex arc. Arc means pathway. So this is a sequential steps of reflex arc. This is very important. Name the connection between two lobes of cerebrum. This is corpus callosum. It is a connector or a tuft of tissue band which connects the two lobe of cerebrum, corpus callosum. Number of cranial nerve and spinal nerve. Cranial nerves are those which are emerging from our brain, and spinal nerves are those which are emerging from our spinal cord. So the number of cranial nerves are 12 pair. And the number of spinal nerves are 31 pair. Examples of cranial nerves are optic nerve, which connects our brain and eye, auditory nerve, which connects our brain and ear, etc. Example of natural and conditional reflex. Natural reflex are present inside our body from birth, and conditional reflex we learn by several applications throughout our life. For example, playing an instrument, solving an numerical problem. These are the example of conditional reflex. And natural reflex is the, we can give example like sneezing, jerking of legs, these are the example of natural reflex. Next question, the three layers of meninges. Meninges is the covering of human brain. It has three layers. The outermost layer is dura mater. The middle layer is arachnoid. And the innermost layer is pia mater. So these three are the layer of the meninges. Next, CSF function and location. CSF is cerebrospinal fluid. Where it is present? It is present inside the ventricles of our brain, inside the spinal cord, and in between the layers of the meninges. That is the in between the layers of dura mater, pia mater, and arachnoid. The fluid CSF will be present, and it acts as an shock absorber, and medium which provides nutrition to the brain cell. Example of a neurotransmitter. This is acetylcholine. Is the example of a neurotransmitter. Nerves connected to eye, ear, and nose. The nerve which connects the eye is optic nerve, which connects our ear, our auditory nerve, and nose, olfactory nerve. Three nerves: optic, which connects our eye, auditory, which connects our which connects our ear, and olfactory, which connects our nose. These three nerves are the example of cranial nerve, which connects our brain to eye, ear, and nose respectively. Next chapter is sense organ. The layer of eye which is richly supplied with blood vessel. We have three layers in our eye. First one is first one is sclera. This is the outermost layer. The middle layer is choroid and the innermost layer is retina. This outermost layer sclera provides protection to the eye. The middle layer choroid, it is richly supplied with blood vessel which will provide nutrition to the eye. And the innermost layer retina, it will contain the photosynthetic cell like rod cells and cone cell. So the layer of eye which richly supplied with blood vessels is choroid. Next, pigment present in rod and cone cell. Rod cells and cone cells are two cells which are photosensitive cells present in the innermost layer of eye, that is retina. Rod cell contains a pigment known as rhodopsin. And cone cell contains the pigment known as iodopsin. Rod cell is responsible for dim light vision or black and white vision, and cone cells are responsible for bright light vision. Lacrimal gland location. Lacrimal glands are located on the upper part of our eyelid here, and these glands are important because it produces tear. Name the enzyme present in tear, that is lysozyme. It kills the pathogen present in the eye surface. Next, what are yellow spot and blind spot? Yellow spots are the region spots are the region present in the retina where the number of rods and cone cells are maximum. This is the region of best vision. And blind spots are the region where no rod or cone cells are present. That is, it is a region of no vision. So, yellow spots and blind spots. Next, myopia and hyperopia cause lens used for correction and diagram. This question is very, very important. Let me draw the diagram first. Here, in myopia, when the light is coming from an object, it will strike on the lens and converges. The light ray converges. As a result, the image is producing just before the retina. So we can use concave lens to cure this problem. On the other hand, in hypermetropia or hyperopia, the light is passing through the lens. When the light is passing through the lens, it is producing the image outside the surface of the retina. And to cure this problem, we use convex lens. So, in the Selena publication book, the diagrams are very good, and normally the diagrams come from that book only. So, this question is very, very important from the part I. Hyperopia and myopia, diagram, cause, and the corrected lens which is used here. Function of organ of Corti, eustachian tube. Organ of Corti are specific type of hearing cell which are present inside the cochlea, the part of inner ear. This organ of Corti receives the signal of sound, becomes activated and produces the stimulus which is sent to the brain by means of auditory nerve. So organ of Corti are responsible for hearing. On the other hand, eustachian tube are a small tubular structure which is connected through our middle ear and throat, and which equalizes the air pressure on the both side of eardrum so that it can freely vibrate. Last question, three bones present in the middle ear. These three bones are malleus, incus, and stapes. The three bones together are known as ear ossicles. These are present in our middle ear. Next chapter. Endocrine system. First question. Give example of a mixed gland. Mixed glands are those which acts both as exocrine and endocrine medium. Exocrine glands means which will secrete enzyme, which are having a specific duct. And endocrine glands are those which will release hormone, which do not have a specific duct. They will release their secretion directly into the blood, and blood will carry this or toward the target organ of our body. So mixed glands are those which acts both as exocrine and endocrine gland, and the examples is pancreas. It's the best example of mixed gland. Other than pancreas, there are two another glands which can act as mixed gland. One is testis, another one is ovary. Why? Because testis produces the hormone testosterone, the male sex hormone, as well as testis is also responsible for sperm production. On the other hand, ovary produces the female hormone, female sex hormone, estrogen, as well as it can produce a female gamete, ovum or egg. So due to this reason, other than pancreas, another mixed glands are testis and ovary. Next question, which gland is called master gland of our body and why? The answer is pituitary gland. Pituitary gland located in our brain, which is hanging from the hypothalamus by means of a stalk or infundibulum. This is known as a master gland of our body. Why? Because pituitary gland produces such kind of hormone that will direct or that will regulate the function of another endocrine glands present in our body. So pituitary gland will secrete some hormone that will direct other endocrine gland to secrete their respective hormone. For example, if the pituitary gland secrete TSH, thyroid stimulating hormone, it will go and switch on the thyroid gland to produce thyroxine. Now, for this reason, pituitary gland is considered as the master gland of the body, and hypothalamus is an extra information. It is considered as the master of master gland because it controls pituitary gland. Pituitary gland controls other glands of our body, and hypothalamus controls pituitary. That's why it is the master of master gland. Next question. All the hormones secreted from the respective gland and their deficiency diseases. A chart is provided in your book. This part is very, very, very important. 4 stars are here. It can come in the form of matching. For example, hyposecretion of insulin results in diabetes mellitus. Hyposecretion of ADH hormone indicates diabetes insipidus. Hypersecretion of thyroxine indicates exophthalmic goiter. Hyposecretion of thyroxine indicates simple goiter, cretinism in children, myxedema in adult. Hypersecretion of growth hormone indicates acromegaly in adult and gigantism in child. And hyposecretion of growth hormone indicates dwarfism. So this respective hormone, their respective gland and their deficiency disease, this part is very, very, very important. Last question from this part, diabetes mellitus versus diabetes insipidus. Diabetes mellitus is caused due to the hyposecretion of insulin hormone. What happens here? Due to consumption of excess glucose, the free glucose are floating inside our body. And the beta cell of pancreas secretes the hormone insulin, which will convert this excess glucose into glycogen and stores inside our body. So if insulin hormone is not sufficient in our body or not producing enough, then the conversion of glucose to glycogen will not take place, and the blood will be loaded with sugar, and this loaded sugar will comes in the form of urine through your body. So this problem is known as diabetes mellitus, caused due to the hyposecretion of insulin hormone. And diabetes insipidus, this is caused due to the hyposecretion of ADH hormone. I have discussed this question in the part of excretory system also. ADH hormone helps in the reabsorption of water. If ADH hormone is less in our body or insufficient in our body, then the reabsorption of water will not take place, and the urine will be with higher concentration of water. Urine will be more diluted. So it will release excess extreme water from our body, leads to diuresis or dehydration. And this problem is known as diabetes insipidus. But in case of diabetes insipidus, there is no sugar in the urine. But in case of diabetes mellitus, the urines are loaded with sugar. Next. Reproductive system. The first question which you can expect from this chapter, that is, what are menarche and menopause? Menarche is the onset of menstrual cycle. As we know that menstrual cycle is a monthly cycle in every female, every primate. The first onset of menstrual cycle at the time of puberty is known as menarche. And menopause is a permanent stoppage of menstrual cycle that happens in the age of 45 to 48 years of age, and menarche is generally 12 to 13 years of age, and this is actually variable. So the onset of menstrual cycle is menarche and permanent stoppage of menstrual cycle is menopause. Next, name the hormone responsible for ovulation. This is LH. LH hormone secreting from the anterior pituitary lobe. It is responsible for ovulation. And on the day 14th of the menstrual cycle, ovulation will take place. This is another important question regarding this topic. Next, the function of placenta. So, what is placenta? It's a cake-like structure present in between the mother and the fetus. And placenta is responsible for transport of nutrient and materials from mother to fetus and removal of the carbon dioxide and waste material from fetus to mother. Along with that, placenta can secrete two hormones that are estrogen and progesterone, but only during the time of pregnancy. So placenta is not an endocrine gland, it's a temporary endocrine gland, you say. During the pregnancy period, it can secrete estrogen and progesterone and another hormone, that is HCG. human chorionic gonadotropin hormone, but normally it is not present inside our body and the endocrine function is not there. But the main function of placenta is to transport of material, food, oxygen from mother to fetus and removal of waste like carbon dioxide, urea and another nitrogenous waste from fetus to mother. Next, role of scrotum in sperm production. As we know, our body temperature is 37 degrees Celsius. But the temperature required for sperm production is 2 to 3 degree lower than the body temperature, that is 33 to 34 degrees Celsius. So if the testis is located inside our abdomen, the temperature will not assist the sperm production. That is why the testes are hanging from our abdominal cavity and present inside a insulating sac known as scrotum or scrotal sac. And this scrotal sac will regulate the temperature of sperm production, that is 2 to 3 degree below than the normal body temperature. Next, what is fertilization? Fertilization is the unite or it is the union of two gamete, male gamete and female gamete, and as a result when zygote is produced. So fertilization is a process through which the male gamete and female gamete unite together to form a zygote. These gametes are haploid in nature, that is N, and the zygote which is produced as a result of fertilization, that is 2N, that is diploid. Amnion and amniotic fluid. When the fetus is present inside the mother womb, the fetus is surrounded by a balloon-like sac. That sac is known as amnion. So what is the function of this? It will protect the fetus from the external injury. And the fluid present between the fetus and the amnion. For example, this is the uterine cavity. Amnion. And here the baby is like this. And the region between amnion and the fetus is filled with the amniotic fluid. So what this amniotic fluid will do? This is the amnion. And this is the amniotic fluid. It will protect the baby from any mechanical shock, any jerking during the pregnancy period. It will act as a shock absorber. By analyzing this amniotic fluid, even we can understand the sex of a child. Next, gestation period in human, that is 9 months or 280 days. The time period on which the fetus remains inside the mother's body, developmental period, that is gestation period. This is 9 months or 280 days. Next, function of vas deferens and prostate gland. Vas deferens are responsible for transporting of the sperm from epididymis to the urethra or penis. And prostate glands are responsible for secreting an alkaline fluid which neutralizes the sperm in the female genital tract. The hormones secreted from the corpus luteum, this is progesterone. Next, what is semen? It's a mixture of sperm, it's a white color fluid, contains mixture of sperm, secretion from seminal vesicle and other glands. It's a milky white fluid which the male ejaculates, containing sperm, secretion of seminal vesicle and other glands. The last question from this part, in which layer of uterus implantation takes place? The answer is endometrium. It is the innermost layer of uterus, endometrium, on which the implantation will take place. So these all are the questions which you can expect from this chapter, reproductive system. Next chapter. Our next chapter is human evolution. First question from this chapter is example of vestigial organ. So first we have to know what is a vestigial organ. Vestigial organs which are present in our body till now, but nowadays these are functionless and these are present inside our body as a rudimentary form. They have no function but just only they are present inside our body. Examples are appendix, vermiform appendix, vermiform appendix. Next, pinna. These are the example of vestigial organ. Next, who proposed the theory of natural selection and survival of the fittest? The theory of natural selection proposed by the scientist Darwin. And survival of the fittest is also proposed by the scientist Darwin. Full name of Lamarck: Jean Baptiste the Monnet Lamarck. Name the moth discussed in industrial melanism. This is Biston bitularia. Scientific name of human. This is the last question from this part. Homo sapiens sapiens. This is the scientific name of human. From this chapter, very less marks comes. So this you can, along with that, you can only just go through the evolution of human. What are the changes through which the modern man arises from the most primitive form. Next chapter. Population. First question from this chapter. What do you mean by the population demography? It is the statistical study of population. Statistical study of population is known as population demography. Next, difference between vasectomy and tubectomy. This is a very important question. These two are the surgical method in human to prevent pregnancy. Vasectomy is a surgical method in male where the vas deferens is cut and ligate to inhibit the transportation of sperm. Whereas in case of tubectomy, the fallopian tube is cut and ligated so that the ovum cannot be transferred. So these two are the surgical method which the female and male applies to prevent pregnancy. Next, deforestation versus afforestation. Deforestation is a cutting down of the trees and afforestation is the implantation of the trees. Deforestation will lead to global warming and afforestation will the rescue method of global warming. Birth rate and death rate. On an 1,000 individual, how many newborns? That is the birth rate. And same on an 1,000 individual, how many deaths? The rate is death rate. Last question from this chapter, MTP, full form of MTP. This is medical termination of pregnancy. Commonly, this is known as abortion. So, MTP is a medical termination of pregnancy. Last chapter, pollution. Example of biodegradable and non-biodegradable waste. So biodegradable waste are those which we can degrade by using microorganisms, whereas non-biodegradable cannot be degraded by means of microorganism. As an example of non-biodegradable waste is DDT, plastic, and biodegradable waste includes vegetable peels, animal excreta, plant residue, etc. Next, these are very common questions which we can expect. Air pollution, its cause, effect and control. How to control the rate of air pollution? What are the several effects of air pollution? For example, breathing problem is there, bronchitis is there, and what are the general cause of air pollution? Water pollution as well as the general cause of water pollution, its effect and control. Example of pesticides. It will kill the pest, DDT. The full form of DDT is dichloro diphenyl trichloroethane. Acid rain, several dangerous acids like H2SO4 or HNO3, which will combine with rainwater and falls down on soil. As a result, the soil becomes acidic, the fertility level decreases, the rate of crop production decreases. Not only that, the acid rain can also hamper the sculpture. The Taj Mahal is, the walls of Taj Mahal is destroyed by this effect of acid rain. So these two are the acid which you can find in the acid rain, that is H2SO4 and HNO3. Next, you can just go through once for greenhouse effect, global warming and ozone hole depletion. These are the general topic. So what are the various way to protect one chemical which is releasing from the refrigerator, that is CFC, chlorofluorocarbon. Chlorofluorocarbon. It is emitted from the refrigerator and that are responsible for greenhouse effect or global warming. So these are the question you can expect from this chapter. Last one is Swachh Bharat Abhijan, its aim and launch date. On which date the Swachh Bharat Abhijan was launched and what is the aim of this Abhijan. So these all are the questions we can expect from this pollution chapter, but these are it carries very less marks. So now here in this slide I have given some important diagram which you can expect from this subject. Nucleotide, nucleosome. This is the from first chapter, cell, chromosome, cell division. All the phases of cell division. This is very important. Any one stage will come in the form of pictorial diagram. It may come. Chromosome, structure of a chromosome. This is very important, showing the centromere. The question may come like that. What is a centromere? So the point of attachment between two chromatidome. Structure of stomata is another important diagram you can expect. Structure of chloroplast. Malpighian corpuscle. It is the structure containing Bowman's capsule and glomerulus. Pathway of reflex arc, which I have already explained. Neurons. Myopia and hypermetropia is a very important diagram you can expect definitely. And the diagram of sperm. This is a part of reproductive system. So along with this diagram, we have come to the end of this discussion. So these are the questions we can just go through once before going to the exam. And at last, I will tell you one thing, that best of luck, try your best, and everything will be good. Thank you.